Welcome to week 2 of CHE124 General Chemistry 2 Laboratory. This week we actually start a project which will last the entire semester. The semester is the investigation of phosphate concentrations in the environment, and so we'll learn the technique to do that. And we'll also learn some other skills. We're going to write a proposal. You guys are going to write a proposal for me to propose what area of the environment that you're going to study. You're going to find that location. It could be Table Rock Lake, could be Taneycomo Lake, could be some stream or creek near your home. All of those are options. And we're going to include some kind of variable, maybe two different streams and a confluence, or where a wastewater treatment plant is, above and below where the confluence from the wastewater treatment plant comes into another system. So if you sampled above and below and where that interface is, then that would be a nice variable. And in that, we're going to look at those phosphate concentrations and if those things affect the phosphate concentrations in the natural world. So this project literally will culminate with a report and poster in the last 2 weeks of the semester. And so anyway, so this is a big project. We actually do a couple other labs connected to it. We do the analysis of phosphate this week in laboratory, the physical experiment that we're going to do. So we can, well, when we write our research proposal, we can put in a methodology that we're going to use to analyze for the phosphate. And so you'll practice that. And then actually next week, we're going to do calibration curves from Beer's Law on another molecule. And so we'll look at it in solution and its concentrations, just so you get a chance to practice building calibration curves. And then the following week, we're actually going to do an analysis of phosphate in fertilizer. And the reason I want to put that lab in is because phosphate in the environment is actually a good thing. It's a mineral, primary nutrient, and that it literally provides food for things that are growing, and without it, we couldn't exist. But it can become a pollutant when it's in excess. And so I want you guys to see it from the perspective that when phosphate is being used in a good way, and then where we might see it as a pollutant. So I'm not trying to make the phosphate ion always negative and a pollutant in that way. So that's why we are doing the next couple labs to learn better to do that. So I'm going to focus this conversation that we're having in this lecture more on the bigger project. I've got a couple slides at the very end of the procedure, but I want you guys to develop your own, and I'm going to guide you as much as you need, and I've given you enough information to analyze for phosphate in this week's laboratory setting. But let's talk a little bit about the bigger project. And so the bigger project is a proposal at the beginning, and I specifically want you to take this lecture set, open it up, and I want you to select this text, or either that or you can do it from the handout, and I want you to copy and paste this into a new Word document. And I want you to use these headings specifically, all right? So I'm putting out a call for proposals, and formatting, I wanted it formatted exactly as this is put together here. So under 1, I want the names of the team members. Under number 2, I need your statement of purpose and focus of your study. And in number 3, I want the importance of the study. I want some sort of justification, 100 to 200 words, something along that line. If it expands past that, that's fine, don't be shorter. All right, so justify why you're looking at, or what the thing is that you're trying to observe in relation to the phosphate concentrations, all right? And you're not going to save the world, I promise, okay? So don't write it in that way. Most of the time we're doing background studies or looking at the relationship of maybe the hatchery to phosphate concentrations, or, as I said, a wastewater treatment plant. And so before and after to see if there's any influence, okay? And so anyway, on this last one, I want detail. I want each one of these ideas from 1 to 6 expanded into at least a full paragraph. That's the minimum, okay? Not one sentence, not one word. I want them expanded, how the known samples will be prepared. So that means how you're going to collect them, what are you going to keep them in, all of those things. The appropriate wavelength for analysis, how you found that. You're going to find that in lab this week. We're going to do a scan of the visible spectrum and look to see where the peak absorbance for the phosphate ion is. And well, actually, when it's interacting with an indicator, we'll talk about that in a minute. And then how the graph will be prepared. So there's a graph that you have to make. It's called a calibration curve. And so I want you to discuss calibration curves, what the relationship is there. Well, you're going to use Beer's law, so that's something I would expect to read about in that statement. Accuracy. Well, in accuracy, one of the things in science is that we compare it to something that we know. So to make our calibration curve, we're going to prepare standards and then compare the absorbance of our unknown to standards. And then we can do repetition to make sure that we're getting good runs and so on. And so anyway, so I want you to discuss that in that way. And then how you're going to do the sample collection, details, all right? And I want the equipment that you're going to need to do the study because we have to look to see if we have it. Now, we actually have some devices which you can do depth studies, surface, 5, 10, 20, up to about 100 feet is what our rope will reach. If you have a way to access the lake, whether that be a dock or a boat, you could do a depth study. That's just a suggestion, so I'll just throw those in as I go when they come to mind. But anyway, I want a proposal for research. Now, to do some of this, we're going to have to do this week's lab. And so when we do this week's lab, you're going to learn. So I want you to take good notes in today's lab to know how the samples are going to be prepared. And I want you to talk about the color indicator and those types of things. And so that's part of the process to learn how to do that. Well, and actually to find out the equipment that you're going to use. So anyway, all right. I'm going to judge your proposals. So they're going to be judged on the following criteria, the proper sections. If you don't do it in the format that I have asked, then it will be rejected automatically and you'll have to rewrite. Okay? The importance of the study and the feasibility or probability that it can be actually completed. If your proposal suggests that you trespass on someone else's property, then I'm going to have to say no. Okay? So if you're going to get a sample from somewhere on private property, then you would have to have permission. Now, being a college student doesn't give you permission. It would have to be asked for through the proper person. And so be aware of that. When you're thinking about picking sampling locations, public access is the best place. Okay, so it's something to think about, something that's available to the general public. All right, so importance of the study. The proper description of the preparation of standards, for example. I'm going to look at that. I'm going to look to see if you actually looked for the correct wavelength during the scan or actually arrived at that. And so if the wavelength's incorrect, I might reject it. If you have appropriate information for graphing, okay? If you don't have enough detail, I'll reject it. And assurance of valid lake samples, proper collection techniques, and those types of things. You might need to actually look some of this stuff up, right, to make sure that you're doing it appropriately. There's things like keeping samples cold. When we do water samples, we can slow the chemistry that's naturally occurring by actually taking our samples, collecting them, triple rinsing the containers, putting them into a cooler, which is cold down to, so we get our samples close to 4 degrees C. And so that slows down the chemistry and helps to keep our concentrations constant. Now, it would be better actually to sample at location, but I don't have enough devices to do it that way, so we'll have to bring our samples back to campus, all right? So anyway, assurance that appropriate safe lab practices will be followed, and so I want you guys to at least acknowledge that, how you're going to be safe. If you're on a boat, you're going to wear life jackets, for example. And so anyway, so please address all of those things in your proposal. You're having some questions right now. All right, what should you study, lake or river? All right, should you look for things that would have high phosphate? And should you look for a variance, whether it be time of day, water depth, before and after some kind of treatment plant or a hatchery or something like that? And then other things that we can talk about is algal growth. If you're giving me that this water is very high in phosphorus, then there should be other things that indicate that, such as a lot of growth of algae and plant life in such a way, even if it's very, very high, there would be something that could be called eutrophication. And so I want you to consider that, right? And how do we accurately determine concentration? How do we determine uncertainty and those types of things? I want you to consider that when you're putting this together. So those are things that you're asking, and I want you guys to investigate as part of the learning of this process. This is just giving you some ideas that are local: Taneycomo, Table Rock, Turkey Creek, Bull Creek, C of O cattle runoff, the dairy stream, the lake honor, dishonor stream. You can get to Taneycomo very easily from campus. So all of those are possible sampling locations. I know with COVID this is going to be kind of strange this year, and we'll have to see. Everything may be on campus this year, but it's possible that you have group members who are commuters, and they may be able to grab off-campus water samples and bring them back for your group to sample. Okay, so anyway, possible variables, I've already mentioned those. Near inlets or confluences above and below them to see if two different streams have an effect on each other, the depth of sample, time of day, and so on. So those are variables that are all possible. The things that I want you guys to work on, educational components, is we're going to work on group work. We're going to work on developing our own procedures instead of having one given to me in an exact format as we normally do in general chemistry labs. All right, so you're going to develop your procedure. You're going to write your procedure that you're going to use specifically and give that to me. All right, and then obviously we're working on our science writing and science presenting, or what we would call science communication. And so we're trying to strengthen you. If you follow on and do more chemistry, we culminate with a course called research. It's a 3-semester course, and the first course mimics this project in writing a proposal and using primary literature to do that. The second semester is actually spent in the laboratory completing the experiments, and then the third semester is spent writing up to develop your own individual full report and poster. And those projects are much more comprehensive than this one. This is an introduction to give you a taste of, if you stay in chemistry, how you'll culminate your career. So anyway, so this is the beginning of that. And all right, a timeline. So a timeline for this project. This week in lab, you're going to complete the phosphate analysis for an unknown sample here. So in other words, you're going to make a calibration curve, and you're going to take that and use the equation from that line, straight line, to find the concentration of an unknown. You're going to form teams. I want groups of 4 to 6 students as teams, and I want you to get together with your team and try to figure out, well, what's your project going to be, to kind of get that started on paper. Okay. First week of February, that's when the research proposal will be due. So you have a couple weeks to write that, to turn that in. I will fail some of them, I will pass some of them. I'll just have to read them to let you know. If I fail it and return it to you, then I'll give you a week to update that and to make the corrections, add the content that I'm looking for, and then resubmit that. And then I'll look at it again, and most likely I'll approve it, because I hopefully will have given you good enough instruction to make those corrections. But sometimes I occasionally will reject it a second time, and I will have you rewrite it and turn it in. All right, and then once those are done, then you're free to do the chemistry as you have planned it. Okay, so you'll collect water samples, bring them back to the laboratory, and sample them. And by the first week of April, really, the lab work should be finishing. Okay, it should be done, so you can start the writing process, and you're going to write a report, and then take that information from the report, condense that, put it onto a poster, and then that poster and report will be due the last week of course. All right, so this week, or this year, that's the last week in April. And so you're going to come to lab, present the poster, and we as a class together, as a research group, will edit that poster so that we can take your work and present it to the public. And I'll collect the lab report and grade those as they are that week. And then during finals week, we have an on-campus poster session. And so the campus community is invited to come see the work that you've done. You'll also see the work of our research students, what they have done in our advanced classes from organic on. Usually do a poster in the spring semester. So you'll be able to see their work. There'll also be posters from biology, possibly engineering, and other fields of study here on campus. So we usually have a good group of people. Hey, there'll be cookies, so something to look forward to. All right. This is an example of the final culminating poster, and so spectrophotometric determination of phosphate in residential and golf course streams. And so it gives an introduction, the methods that were completed, the materials that were used, a conclusion to their data, and then the references that they got their information from for each parts of this project. This is a calibration curve, all right, made from Beer's law. It's absorbance versus concentration. Now absorbance, this is an incorrect thing here. Absorbance has no units, so it's not in nanometers, just to let you know that. This is a picture showing the sampling locations, actually giving some coordinates related to those. And I think there should be coordinates for each location. And then the concentration, so these are the results in table form to make them easy to see. And what you find is that a golf course has a high influence on the amount of phosphate. If I remember right reading this, there was some rain involved, and so probably some runoff from a residential area that was also influencing that. So anyway, the project. This is how it will culminate. It will also, there will be a report. And so just to let you know, I also brought up an example of, a proposal. And so just to show you one that it is literally formatted, this is College of the Ozarks Waterways, and the team members' names have been removed from this, but the statement of purpose, and then so in that statement they also said where they were going to sample. They gave the locations that they were going to sample, and so you guys can see those on the map, and the importance of the study. So they made an argument about that and what the concentrations that they were expecting were at. So they did some literature work there, and how the samples will be prepared. And if you notice, this is in detail. It goes through how each standard would be calculated, those types of things. It gave the concentrations of the standards and so on, and ultimately the wavelength for analysis, how the graph will be prepared. I think that needs more detail. The accuracy of the method, a discussion of what they thought was accuracy, how the sample will be collected, and how the samples will be tested. And so, and then lastly, the equipment. You notice that list is kind of extensive. So anyway, I want you guys to come up with the same thing, and then ultimately end where you are looking at the final poster. And there's a document on Campus Web, how to write a report. I want you to follow that format for the report, even in terms of the way things are referenced and so on. It's very specific. So, all right, that's the project. Today's lab, you're given an experiment to do, and you're going to do an analysis of phosphate. Phosphate is not colored in water. You can't see it, so we add a reagent, right? Molybdophosphoric acid, and that forms a complex with our phosphate, and this has a purple color. And so we can see that, well, and we're going to use the eyes of the spectrometer to see that. And so you're going to build a calibration curve which uses Beer's law relationship. The absorbance is equal to the extinction coefficient times the cell width times the concentration. And so these two are constant, so that means that absorbance is proportional to concentration. And so we will see a color change when phosphate is present. if you printed your lab early, this has changed a little bit. I say to make at least 5 samples from 0.01 to 0.6 part per million range, and then you'll need a blank, and you can do the concentrations of your standards from a 2 part per million standard solution. We're going to supply that, and so you'll need to do dilutions. Once you get those diluted, you're going to take 10 milliliter samples of each and put them in a 100 milliliter beaker. You're going to add the color reagent. You're going to swirl those beakers. And actually for those, this says 15 minutes on the slide. That is incorrect. You only need 5 minutes or less for those to develop. So you can shorten that time a little bit. That should be corrected in your lab handout now. But anyway, you're going to take a cuvette, put an amount of your sample in that. You're going to measure the absorbance of that sample. You're going to measure the absorbance of all of your standards. You're going to build a calibration curve with the standards and then compare your absorbance of the unknown to the absorbance found in the standards. And from that, you're going to be able to calculate the amount of unknown that was in that sample. So anyway, or the amount of phosphate in that unknown sample. All right, so that's the project that you're going to do today. I'm going to let you guys fill in the details on that when you're writing your own procedure. So you should be working on writing that procedure for your long-term project as you're doing today's lab. Okay? So anyway, a useful equation. We used it last week in lab. We're going to use it again this week. C1V1 equals C2V2. And wear your eye protection when you're working on your experiments. So if you have any questions, let me know at the beginning of the lab. I'm looking forward to this. This is one of the things that I really enjoy about general chemistry, this project, and reading some of your ideas. And so I'll see you in lab this week. Have a great day.